It's like those memes, like, no one, Mr. Blackburn, likes sperm. Like, yeah, so you don't need to know for the AP exam, like, the exact anatomy of a sperm, but I'm building towards, you need to understand how to interpret a pedigree based on mitochondrial DNA. And that's not something your textbook actually went over. So that's why I'm showing you the structure of a sperm. To understand that, you need to understand the basic anatomy of a sperm. So sperm's kind of interesting because basically the sperm, it'll essentially have a part where like you just have your nucleus, and then they essentially will like send out all of their mitochondria and concentrate them in an area they call the collar. And why would they do that? Why would the mitochondria no longer be used to do like, essentially like the sperm is no longer just doing like really making a whole lot of things. They're just kind of like, all right, we're just sending all of our energy towards our tail. And why would that make sense for a sperm? What is the job of a sperm? Oh.
To fertilize the egg.
To fertilize that egg, right? So you want all the mitochondria on that tail because you're competing with a bunch of other sperm for that egg, right? So that's why all the mitochondria are focused right here. And so then what would happen is the first sperm to get to the egg and, bless you, and like there could be multiple sperm trying to get through to this egg. At the very tip of the sperm, there'll be, they're called an acrosome. And there are hydrolytic enzymes there. Now, you don't even know what an acrosome is, but what is a hydrolytic enzyme? You need to know what those are. Think back to Unit 1. We talked about hydrolysis. So hydrolytic enzymes do hydrolysis. What were you doing in hydrolysis? Yeah, say. You're breaking things down. Yeah, hydrolysis, you add a water molecule to break down those biomolecules. So that's what the sperm is trying to do here. It's releasing these enzymes that break down the outer portion of the egg called the zona pellucida. And eventually the idea is the sperm will eventually get through into the egg, and then it's only, this is the key part, it's only going to contribute just the nucleus of itself. In other words, only the mitochondria that the sperm used to get to the egg, that all of the mitochondrial DNA that's inside the mitochondria. Remember, mitochondria and chloroplasts have their own DNA. That DNA from the mitochondria, the sperm, does not make it inside the egg. And that's crucial because that means then that only the mitochondria from the female are going to contribute that information. Another point is that in fertilization, all of the nutrients and ribosomes and informational molecules, that would be like your signaling molecules that you'll need to get, they call it a zygote whenever it's like a one-cell embryo before it's done any division. They call that a zygote. The signaling molecules to get that zygote to begin growing and all that, the early stages of development. Basically, all the sperm contributes is just the DNA. Everything else is coming from the female. Okay, that's why the egg is so big. It's like kind of got that storehouse of nutrients to fuel early development before you get the placenta that's attached. Okay, now all of that is building towards understanding this pedigree here. So let's say it's not as common because there's not as many genes in the mitochondria compared to like the overall genome. But there are some genes that are essential in the mitochondria where if you have a faulty gene, you'll end up with a mitochondrial linked disorder. And because all of the mitochondrial DNA comes from the mother, if the mother has that disorder, has that faulty gene in her mitochondria, she will pass on that faulty gene to 100% of her offspring. That's why both are colored. So that's the first important point. Second important point is if a male then inherits a mitochondrial gene, that male, 0% of his offspring will have that. So why is that? Yeah, all right, yeah. All of his faulty, like that doesn't get into the egg, right? Only this. So the female right here, she's the one passing on the mitochondrial DNA. Okay, so that's they a few years ago they had like a free response question based on a mitochondrial DNA pedigree. So just want to make sure you understood that. Last thing is there's only one allele because there's only one set of all the mitochondrial DNA just from the female. That's why there's no dominant or recessive. There's only one copy. Okay. Other than that, you interpret it kind of like a normal pedigree would. Okay. So the last thing that we've talked about these, but I feel like I worry it's kind of fallen through the cracks. Is the two laws of Mendel that you need to know: the law of segregation, and the law of independent assortment. So the law of segregation says that the sister chromatids will segregate independently. What I mean by that is if you're analyzing one gene, let's say we call that gene A, whenever that gene is making the offspring, this is why when you do the Punnett square, like only these alleles segregate independent of each other. This is why, like let's say you have two parents being crossed together, this is why when you do the Punnett square, the law of segregation, this is why there's only one letter here and one letter there. That's what the law of segregation is saying. The alleles segregate independent of each other. Okay? You don't get one, there's not like one parent that has two copies of that gene. Okay? Law of independent assortment is related to the law of segregation, but it's analyzing the homologous chromosomes. So this law of independent assortment is saying the homologous chromosomes separate independent of each other. So independently. In other words, the copy of a one gene you get from mom segregates differently from the copy from dad. So like, for example, you had like big A, big B, little b, just because like let's say this one came from mom and this one came from mom also. Whenever you're making the gametes, you get 4 possible gametes. In other words, yeah, you might get a gamete that has both of the copies from mom, but they don't always travel together. So you get 4 possible gametes. You could get one big A, little b, another little a, big B, another little a, little b. If they traveled together, you'd only have 2 possible gametes. You'd have big A, big B, and then just little a, little b. If the dad wants something to do this. So if law of independent assortment did not happen, you'd only have these 2 possible gametes. But because they separate independently of each other, you have an equal likely chance of getting a combination of the 2 parents. That's what that's trying to say. Okay, just want to make sure I went over that again. Let me stop the recording.